members, today we are back in our biology, and today our emphasis is going to be on what we are calling practical biology, and I'm going to consider what we are calling physiology. Physiology, mainly, we can look at what we call food tests, we can look at processes like diffusion, osmosis, but today my idea is going to be at what we are calling food tests. Food tests, there are some components of food which we need to discover, but by investigating them or discovering them practically. So, there are some specific reagents which we use when we are carrying out food tests. And some of the reagents, I'm going to show you those reagents as we are beginning. One of the reagents which we are using when we are carrying out food test is what we are calling iodine. When you look at our iodine we can say it is brown in color, or we can say it is yellow. So it depends on the concentration. Now when you look at ours, it is brown in color. We are going to see that this one we are using it when we are testing the presence of starch. Presence or absence of starch. Then another reagent which we always look at or we use when we are carrying out food tests, we have a reagent known as Benedict. This is Benedict's solution. When you look at it, it is blue in color. So when you look at our Benedict, this one is mainly used when we are testing for the presence of sugars. But specifically, we can use Benedict when we are carrying out reducing sugar test as well as non-reducing sugar test. This one we are going to see. We are going to see it as we are moving ahead. Another reagent which is crucial, most of you enjoy vitamins. So one of the vitamins which is important in our body is vitamin C, like in oranges and the pineapples. So we have what we call DCP-IP. You can see that this one is a deep blue. When you compare this blue and this blue, when you compare the blue, this one of Benedict, and the blue of DCP-IP, you will discover this is the deep blue, and this one, it is just pale blue. You can see that blue is pale. Then another reagent which is very, very important is what we are calling copper-2 sulfate. Copper-2 sulfate here is important. It is also light blue, as we are seeing. Somehow resemble the other one of Benedict, but this one light blue. So this one we are going to use it in combination with another reagent which is vital, and this reagent we are calling it sodium Sodium hydroxide. So you can see our sodium hydroxide is working together with the copper-2 sulfate. Now these two reagents, we are going to use them when we are carrying out what we call protein test. And now these ones are going to give us what we are calling the Bioretiz test. Now this one, vitamin C test, DCP-IP. Then this one is starch test. So this one, it is for starch. That one we are calling starch test or iodine test. Then we have this one here, Benedict. As you can see, our Benedict. This Benedict we are saying is for reducing sugars as well as non-reducing sugars. But we are going to see how we treat it when we are investigating reducing sugars as well as non-reducing sugars. So with this one we are going to use it like in Benedict's test or reducing sugar test or non-reducing sugar test. Now from there we have another reagent which is vital. It is appearing colorless. This one, hydrochloric acid or HCl. This one we are going to use it when we are carrying out non-reducing sugar test. Don't forget that. This one is very, very important because it's going to help us to do what we call hydrolysis or breakdown of non-reducing sugar, as we are going to see as we are moving ahead. Grasping properly the reagents and their colors. You have to master the reagents and their colors. Finally, I'm saying DCP-IP, deep blue, vitamin C. Then iodine, brown, or you can even say yellow, that one, starch. Then copper-2 sulfate, pale blue, that is proteins. But remember we have said in a combination with sodium hydroxide. Benedict, sugars, specifically reducing and non-reducing sugars. HCl, we shall see it in non-reducing sugars. Take note of those colors. Now, at the start, we are going to look at starch test. Starch test, we are going now to consider our reagent, that is iodine. That is one we are going to consider as our main reagent. Now, as we are considering our iodine, we have to look at the sources of starch, which we are going to test for. Now, today for me, I'm going to use my solution. This is my solution. But that solution, I've made it by using this food which we enjoy a lot. You can tell the food that is our flour, where we are getting our portion. But remember, it's the same in the cassava, the same in the millet, and so on. So this is my sauce, which I mix to get this one. First of all, we are going to look at what the color of this one here before we test. When you are carrying out food test and you are recording, you record the initial color, that is the beginning color, and then the final what? The final color. This one, we can say that this solution is turbid. The solution is purely what? Turbid. You can see that turbidity. So we say turbid solution. When you are recording, we shall turbid solution. Then this one, of course, it is brown as we are seeing. Now I'm going to obtain my test tube. But remember, when you're writing procedure for this one, they will say to 1 cubic centimeter of solution S, add drops, specifically they can be 3 drops, add 3 drops of iodine what? Iodine solution. That will be our procedure. So how are we going to measure our 1 cubic centimeter? Then we get a small measuring cylinder, this one here. That one is a 10 mm measuring what? Cylinder. So what do? We get our measuring cylinder, then we pour there our solution. Pour there our solution. We are going to get our measuring cylinder, then we pour there our solution properly. We said when you are pouring, you make sure that you don't wet your working what? Your working place. You can even use the 4 cubic centimeters. It depends. Then, if for example, I'm going to take my measurement there as my 1 cubic centimeter, then now I get my test tube, then I put my, remember to already test tube properly, as I always tell you. So you put there your solution. That is our solution. Then after putting our solution, turbid what? Turbid solution. Then we are going to move to the reagent. Our reagent is here. Don't forget our reagent. This is our reagent iodine, brown. Then we are going to get our brown iodine. Then slowly, we put there drop by drop. The man said, put 1, then we put 2. But before I even add, I shake a bit. You can see that my solution, which was turbid, is now turning darker blue. Or you can even say black. But avoid saying blue, black. Even if you add excess, you can see. Now, this one is going to become black solution, or you can even say darker blue solution. And our observation is going to become turbid solution. Turbid solution, this one here, turns to a darker blue solution. And then our conclusion will be starch present. Avoid using chemistry language. Confirm, you can just say starch present. Now, if we are going to do a negative test there, I can do a negative test to show you that the reagent doesn't turn every time darker what? Darker blue. We can use now a negative test here using water. When I get my water, you can see your water. Then I put my solution like that, which is my water. Already you know it. Then after doing that, I also transfer the same iodine as I've been what? Doing. So I get my iodine. Members, I was saying that we can carry out a test which is going to give us negative results. We have used the water here. Then we are going to add iodine and we see whether we shall get the same darker blue solution. Then I get my iodine. I put the first drop. I put there. You can see that this solution, which doesn't have our starch, for it is just turning yellow. So meaning when I'm recording this one for water now, which I've used, I will say colorless solution turns to a yellow solution. And there I say starch absent. But this one here, the first one, this one here, which we had, you can look at ours here, which had starch, so we can say that the turbid solution turns to a black solution, a darker blue solution. You always write one color, don't stroke, darker blue, then blue, black, what? That is rubbish in what? The science. Always write what you see exactly. Now members, when we put that one aside, our second test is going to be on what we call the proteins. Now proteins, let me even put this one aside. Our proteins, we said, the common test is what we are calling the Bioretiz what? Bioretiz test. Bioretiz test is a combination of copper 2 sulfate and another reagent which we are calling sodium hydroxide. So when you bring the two, they will give us the test which we are going to talk about. Now, sources of proteins, you always enjoy meat, you always enjoy fish, you always enjoy egg. Now you can look at our egg there. This is now, that one is going to act as our source of proteins. Now I'm going to just I'm just going to break now. Then I get a solution from our egg. Then from there, we shall now see what happens next. Now I get my solution from my egg like that. Then I'm going to add water. Now I get water, I add some water there. This water here now, I can even add water. Let me try to add some water, as you are going to see. Add some water there, like that. Then afterwards, we are going to do what? Stir. So as we are stirring our solution, now we are going to discover that our solution we have obtained is like that. Now members, this solution here is our solution which we are going to call out our protein what? Our protein test. When you look at that solution, I've now called it solution P. My solution P. Now this is my resultant solution after putting my egg albumin. So egg albumin is one of the source of proteins. Now I can label it solution what? Solution P. Now my solution P, now I'm going to call out what we are calling Bioretiz test or protein what? Test. What am I going to do here? I'm going to measure 1 cubic centimeter. Always they say here, measure 1 cubic centimeter of solution P. This one here, then I get my measuring cylinder. The moment you say what measure, then you get your measuring cylinder, then you pour your solution properly. That is my solution now. Which solution is coming from where? Now, I've measured 2 cubic centimeters of my solution. Then I'm going to add it in my test tube. After adding it there, remember as when you are using the test solution, always use little, don't bring a lot. A lot will give you wrong results. After doing that, I'm going to add there 1 cubic centimeter of. Now this is our solution. When you look at our solution, members, guys, when you look at the solution here, this solution is series turbid. Never be confused. So we can say turbid solution. Even this one is turbid. To our turbid solution, we are going to add there 1 cubic centimeter of sodium hydroxide. This is our sodium. Our turbid solution, guys, we are going to add sodium hydroxide here. Remember I told you Bioretiz test. We first add sodium hydroxide. You already set 1 cubic centimeter of the solution. Add 1 cubic centimeter of sodium hydroxide. Then you add drops. Preferably 3 drops of copper 2 sulfate can be best. Then we do what? We shake. Now I put my first drop. I can do some shaking, but you can see there is some color which is beginning to come there, some strange color. Then I add more, then I shake. Now when I shake, members, you can see what we are getting. This one is what we are calling purple. Purple now. That is the purple solution now. So what are we going to write as our observation, members? We are going to write now that a turbid solution, we are going to write that a turbid solution, this one was our turbid solution, our solution P, turns to a purple solution. If you don't say purple, you can say violet, but don't stroke them, purple stroke violet. You pick one. Now, this one, our conclusion is going to be our deduction, proteins present. Remember, proteins can also be tested by using million. But for me, I've carried out this one for Bioretiz. Now, don't forget that proteins can be in eggs. We have used an egg. The meat you enjoy, there. Cowpea, milk, and so on. Everything which I've mentioned there contains proteins. Now we are going to call out our negative test now. Assuming I use a solution for starch. The other one which we used for starch, that was our solution S which we made. That was solution S, which gave us good results for starch. Then let me try to use it and you see whether we can get the same observation. Now I put my solution S like that. I've not measured using a measuring cylinder. Then I also put sodium hydroxide. Put sodium hydroxide. Putting sodium hydroxide. Then what are we going to add? Guess it. We are going to add copper 2 sulfate. Now I get my copper 2 sulfate. I put some drops. 1, 2, 3, 4. Let me do the shaking. Now members, try to be observant. When we use the solution P, our results were like this. When you added the sodium hydroxide, the copper 2 sulfate, purple solution or violet solution. When we have used our starch solution, which we confirmed already, we are seeing that the solution is mainly like this. This is now blue. This is blue. But this is what? Violet. This one we say proteins absent. Here we say proteins present. That is after adding sodium hydroxide as well as copper 2 sulfate. Members, take note of the procedure properly. It is sodium hydroxide first, then drops of copper 2 sulfate next. If you interchange, you are going to obtain opposite what? Results. Let us try it. So let me get my solution P, which gave us this one. Don't forget. Now let me get it again. Now I put it in my test tube. That same solution. P. Don't forget that the one with the proteins. Then I add now copper 2 sulfate first. Let me do the opposite now. Let me do the opposite. Now I'm adding copper 2 sulfate first. I'm adding copper 2 sulfate first. That is the one I've added. Then I'm adding sodium hydroxide last. I'm interchanging the procedure to show you what is going to happen. Then I add my sodium hydroxide. Now when I shake properly, you can see now, you get this one, get this one, the one where we have reversed the procedure, and this one is the right procedure. Here you are going to obtain blue precipitates when you have reversed the procedure. When you have used the correct procedure, we are getting violet solution or purple solution. So take a note, never reverse, and then you put copper 2 sulfate first. It should be sodium hydroxide what? First, for better results. Now, the other test, I think we are done now, that one. We are going to look at what we call a vitamin C test. Don't forget that we say our chemical which is going to work for us there, we are calling it DCPIP, or some biologists can say dichloropheno, endorphino. But now, what we are going to do here, we are going to obtain our vitamin C from one of the fruit which you always enjoy when you go to some parties. So now we have our lemon fruit here, but remember even in pineapple and many other fruits, we may get vitamin C. So I'm just going to cut my lemon, then I get my juice here. So I get my juice. Let me try to squeeze and I get some juice from my orange, from my lemon. So let me squeeze a bit. There we go. I have got my juice from my lemon fruit. Then after getting my juice, now I'm going to transfer my juice into a test tube. Now I put my juice into a test tube. That is my juice from the lemon. After I'm getting my juice from there, now I'm going to get a clean test tube. And then I transfer 1 cubic centimeter of my DCPIP. Now, when you look at this test, it is the only test where you are going to add the test solution. The test solution, that is the food you are going to test, to the reagent. The other ones you have been adding the reagent to the food. Now here we are adding the food to the what? The reagent. So I get my DCPIP. Remember we said it is deep what? Deep blue. as you are seeing. Now what am I going to do? I'm going to transfer now some drops of the food. I'm going to transfer some drops of the food now. So there I get my food. This is the food or the juice which you have got from the lemon. So I get my dropper. Then I get some juice from my test solution. Then I'm going to transfer drops of this juice. Now I put my drops of test solution. You can see as I'm putting now, you are seeing the deep blue is disappearing. There we go. You can see that now our deep blue solution of the DCPIP is being decolorized. In simple terms, here we are going to record that the deep blue solution turns to a colorless solution. What are we referring to? Remember our reagent, if we are going to put my reagent here, which I have used, that is DCPIP. You can see members, the reagent. And then when I added the juice, this is when I added the juice. Let me try to repeat and you see. I add my juice. The deep blue does what? Disappears. So we can record here that the deep blue solution turns to a colorless solution. That will be the base. But in some books you'll see someone telling you the deep blue solution is decolorized. But the basic way of recording it will be the deep blue solution turns to a colorless what? Colorless solution. And that one means vitamin C is what? Is present. We have got it from our lemon.